Assalamualaikum and very good morning, girls. Okay, so let's continue our lesson today with the 11.2 dihybrid inheritance. Yeah, dihybrid inheritance. Okay, in the previous class, we already cover 11.1. Yeah, which is referred to the monohybrid inheritance. Okay, so for today, we are going to discuss dihybrid inheritance. Okay, let's go to the learning standard that you have to cover for today. Okay, number one, describe the meaning of the dihybrid cross. Yeah, dihybrid cross. And number two, explain a dihybrid cross based on the Mendel's experiment. And number three, construct a dihybrid cross diagram. Okay, you have to learn how to membina. Construct a dihybrid cross diagram. And number four, determine the genotypic and phenotypic ratios of filial generations in a dihybrid cross. And last one, describe Mendel's second law. Okay, so that is referred to the learning standards that you have to understand for today. Okay, we go to the introduction of the dihybrid inheritance. Okay, so what is a dihybrid inheritance? Okay, di refer to the two, right? Okay, two. So dihybrid inheritance involve inheritance of two characteristics. Yeah, inheritance of two characteristics. Okay, so dia melibatkan perwarisan dua ciri. Yeah, involve two characteristics. Each characteristic is controlled by different gene located at different locus. So when it involves two characteristics, so it will involve two different genes. And these two different genes located at two different locus. Okay, that is referred to the dihybrid inheritance, which involve the inheritance of two characteristics. Okay, figure in the next slide shows a schematic diagram of a dihybrid inheritance between two purebred parents to study the characteristics of P, namely seed color and also seed shape. Okay, so you can also refer to your textbook on page 238. Okay, there is a schematic diagram of a dihybrid inheritance that between two purebred plants. Yeah, okay, two purebred plants for seed shape and also for the seed color of the pea plant. Okay, then the two purebred parents either have a pair of homozygous dominant alleles or a pair of homozygous recessive alleles. Okay, so do you still remember what is a homozygous dominant alleles and also what is a homozygous recessive alleles? Okay, please go again. What is the meaning of the terms? Alright, okay. Then, fertilization between the two purebred parents, fertilization between the two purebred parents produce offspring. Dia menghasilkan anak in F1 generation, or we call as first filial generation, with dominant traits of round and yellow seed. Okay, so let's take a look on the schematic diagram of the dihybrid inheritance for seed shape and seed color of the pea plant in your textbook on page 238. Okay, so this refers to the dihybrid cross. Dihybrid cross between the two purebred plants. Between the two purebred plants that involve a pair of the homozygous dominant alleles. And also the purebred plant of the homozygous recessive alleles. Alright, so how to construct a schematic diagram? Okay, so we are going to discuss how to construct a dihybrid schematic diagram of the dihybrid cross. Okay, so please pay your attention. Alright. Okay, so this is referred to the schematic diagram. Yes, schematic diagram. Okay, before you construct your own schematic diagram, okay, make sure you understand the key. Alright, the key here, right, that is referred to the key that represent the allele. Okay, actually you can choose any letter to represent the allele. So in the textbook, okay, the key given to you, big B or capital letter B represent the dominant allele for the round seed. Yeah, for the round seed. And then the small b represent the recessive allele for the constricted seed. Yeah, small b represent the recessive allele for constricted seed. And the big K, yeah, big K or the capital letter K represent the dominant allele for the yellow seed. And the small k represent the recessive allele for the green seed. Okay, that is the key represent for the allele. Okay, so to construct a schematic diagram, you have to start with the parental phenotype. Yeah, parental phenotype. So actually, what is a phenotype? Phenotype refer to the physical appearance or refer to the characteristic that we can observe, right? So the parental phenotype of the two purebred plants, yeah, which are round and yellow seed. Okay, another one is constricted and green seed. Right, so this is referred to the parental phenotype. And then second one, you have to write parental genotype. Parental genotype, the genetic composition of the parent, yeah, of the two purebred parent. Okay, and the first one here is the genetic composition or the genotype of the brown and yellow seeds is big B, big B, big K, big K. Yeah, so they are homozygous dominant alleles. Okay, I repeat, big B, big B, big K, big K. And then for the constricted and green seed, a purebred plant, okay, the genotype is small b, small b, small k, small k. So that this is referred to the homozygous recessive alleles, right? And then, okay, so in the formation of the gamete, okay, in the formation of the gamete, meiosis occurs. So you have to write here meiosis. Okay, refer to the meiosis occur. Yeah, meiosis occur here and produce gamete, not produce gametes, right? So the gamete, again, the gamete produce for the round and yellow seed. All the gamete consist of the big B, big K. Yeah, and then for the constricted and green seed, okay, the gamete produce is small b, small k. Right? Okay, and then what happened after that? Fertilization occur between the gametes. Yeah, fertilization occur between the gametes and produce F1 generation. Okay, first of all, you have to write first filial genotype or F1 genotype. So, the F1 genotype, all the F1 generation produced are the same. Right? So all the genotype is the same, which is big B, small b, big K, small k. And the phenotype for the F1 generation are all round and yellow seed. Okay, so why all round and yellow seed? Because this is referred to the heterozygous dominant. Yeah, big B, small b. So it will show the dominant trait. Here, what are the dominant traits here? Round seed. And then for the big K and small k here, also heterozygous, and it will show the dominant trait here, which is yellow seed. Alright, okay, so we go to the explanation here. Seed shape characteristic is controlled by a pair of the big B, small b alleles, and seed color is controlled by a pair of big K, small k alleles. So only the dominant traits, namely round and yellow, are observed. Alright, okay, so why they are all round and yellow seed? Because big B is a dominant allele for the round seed, right? And then why the all of them are yellow seed? Because this big K is a dominant allele for the uh, yellow, for the seed, for the color of the seed. Okay, so that's why it will show all round and yellow seed. Alright, and then okay, after that, what happened? Self-cross F1 generation occur. Yeah, okay. So offspring of the F1 generation if is self-cross, dikajuk sesama sendiri. Okay, so here you have to write self-cross F1 generation, and then you have to write F1 phenotype. First filial generation phenotype. Okay, so what is a phenotype? Round and yellow seed, yeah, for the first plant, and the second one is round and yellow seed for the second plant. Alright. So and then okay, you have to write the F1 genotype, the genetic composition of the first filial generation. First plant is a big B, small b, big K and small k. And the second plant is big B, a small b, big K and small k. And then in the formation of the gamete, okay, what process occur here? The meiosis occur here, yeah, and producing the gametes. So these are referred to the possibilities of the gamete produced. Okay, so what are the gametes produced? For the first one here is big K, big B, big K, and big B, small b, big K, and small b, small k. Alright, so these are the gametes, yeah, the gametes producing after meiosis occur. And also for the second one, for the second plant, also big B, big K, big B, small b, big K, and small b, small k. Alright, okay, so what can we summarize here? During the formation of the gamete, any allele for the seed shape can pair with any allele for seed color. Alright, okay, I repeat. So during the formation of the gametes, any allele for the seed shape, yeah, any allele for the seed shape can pair with any allele of the seed color. So for example, the allele for the seed shape is big B, yeah, big B, and also the allele for the seed color is big K here. So the allele for the seed shape can pair with any allele for the seed color, yeah, as you can see from here. So the big B possible to pair with the big K, and big B also possible to pair with the small k. So that is referred to the theory of the Mendel's second law here, right? And then okay, after that what happened? When the F1 generation produced from dihybrid cross in the figure was self-cross. Okay, so what happened? The 16 F2 genotype combination are produced. Yes, 16 F2 or the second filial generation of the genotype composition, genotype combination produced. Okay, so later we will look at the schematic diagram. Okay, the cross shows four phenotype characteristics in the F2 gene generation. Okay, I repeat, the cross shows four phenotypic characteristics in F2 generation. So it's different from the F1 generation. In F1 generation, it's only show two phenotypic characteristics, but in F2 generation, it shows four phenotypic characteristics. Right? And the outcome of the dihybrid cross is represented in a Punnett square. Yeah, outcome of the dihybrid cross is presented in a Punnett square as shown in the figure on the next slide. Alright, okay, let's go to the Punnett square. Yeah, that present the F2 generation. Yeah, the offspring of the F2 generation. Okay, so the hybrid cross of the F1 generation. Okay, so we self-cross the F1 generation. Yeah, we self-cross the F1 generation. Okay, and then by using, so you have to use the Punnett square. Okay, why we have to use the Punnett square? Because there are so many gametes here. So many gametes here for the male gamete. Yeah, for the male gamete or for the for the first plant here. Okay, so there are four different gametes. Yeah, that represent the four different genotype. And the second plants also here also got four different genotypes. Okay, so when we do the dihybrid cross of the F1 generation by using the Punnett square, so what we get? Yeah, what we get? Okay, so we go to the Punnett square here. Okay, the combination, yeah, or the fertilization between the gamete of the big B and big K, yeah, the fertilization between the big B, big K, and also with the big B, big K will produce big B, big B, big K, big K. That is referred to the round and yellow seed. Yeah, okay. So if the gamete is big B, small k, yeah, big B, small k, fertilized with a big B, small k, it will produce. Okay, so it will produce a gamete with a offspring with a big B, small k, small k. So the offspring will have round, okay, round seed and green color. Okay, round. Okay, why it is round? Because the dominant alleles here. Yeah, the dominant alleles that represented by the big P. Yeah, that show the dominant trait which is round. Okay, why the color is green? Because the presence of the recessive alleles. Yeah, recessive alleles. That is homozygous recessive alleles, which is small k, small k. So it will show the recessive traits. Alright, okay. So I choose another one example. If the gamete has small b, small k, fertilized with a small b, big K, okay, so it will produce the offspring, the offspring which has a small b, a big K, small k. Alright, okay, and then what is the phenotype shown by the seed? It is constricted. Okay, why constricted? Because this is homozygous recessive alleles for the seed shape. Alright, okay, and then this one, there's a big K and small k. So it will show the dominant trait because the presence of the dominant allele here, and it will show the yellow color of the seed. Alright, so the phenotype of the seed is constricted and yellow seed. Okay, so what we summarize from here. Okay, yeah? So the hybrid cross of F1 generation using Punnett square will show, okay, will show the phenotypic ratio. Okay, will show the phenotypic ratio, which is 9 round yellow seed. Okay, 9 which are round and yellow seed, 3 which are round and green seed, 3 which are constricted and yellow seed, and 1 which is constricted and green seed. Okay, that is referred to the phenotypic ratio, yeah, of the F2 generation. Yeah, of the F2 generation. So F2 generation is produced when the F1 generation is self-crossed. And then we have to use the Punnett square, yeah, Punnett square to determine the F2 generation. So what is the phenotypic ratio of the F2 generation? 9 is round and yellow seed, 3 are round and yellow green seed, okay, 3 constricted and yellow seed, and 1 is constricted and green seed. Alright, so that is referred to the phenotypic ratio produced in F2 generation. Okay, so this is another example of the dihybrid inheritance, yeah, that involve two purebred plants, yeah, yeah, which are tall and red plants, yeah, and also short and white plants, yeah, short and white color of the flower. Alright, okay, so this is referred to the parental genotype. Okay, this is referred to the parental phenotype. Okay, and then in the formation of the gametes, yeah, what happened? The meiosis occur and produce the gamete here. Okay, and the gamete produce here fertilize, yeah, fertilize and form the F1 generation. Yeah, form the F. Okay, so the fertilization of the gametes, okay, will produce first video generation. Yeah, first video generation or F1 generation, which show the same phenotype, okay, which is tall and red plant, and same genotype also, which are all big T, small t, big R, small r. Right? Okay, so that is the first, that is referred to the F1 generation. And then what happened? The F1 self-pollination occur, or F1 self-cross occur. So in the F1 self-cross, yeah, in the first video generation, when it is a self-cross, okay, so it will produce the second video generation. So it will produce the second video generation. Alright, so how to determine the second video generation? We have to use the Punnett square. So this is referred to the Punnett square. Alright, and then you have to determine, yeah, determine the offspring produced here. Alright, so at the end, okay, we will get the phenotype ratio here, okay, which are 9 are tall and red color of the flower, 3 are tall and white color of flower, and 3 which is short and red color of the flower, 1 which is short and white color of the flower. So it will show the phenotype ratio 9, 3, 3, 1. Yeah, alright. Okay, so that is referred to the dihybrid inheritance. Okay, make sure you can know the phenotype ratio of in the dihybrid inheritance, which is 9, 3, 3, 1. So how to determine the F2 generation, we have to use the Punnett square. Yeah, alright. So, dihybrid inheritance will relate with the Mendel's second law. Yeah, okay, so dihybrid inheritance that we have just discussed now, that is referred to the Mendel's second law. Or we can, we can say that law of independent assortment. Okay, so this law is introduced by the Mendels. Yeah, by the Mendels. So what is stated by the law of independent assortment? Yeah, law of independent assortment. So it is stated that during the gamete formation, each member of a pair of allele can combine randomly with any allele from another pair of allele. Right? Can I repeat? During the gamete formation, each member of a pair of allele, can combine randomly with any allele from another pair of allele. As we discussed in dihybrid inheritance of the schematic diagram for the seed shape and seed color of the pea plant on the page 238. Okay, what can we say? What can we summarize from the schematic diagram? So please look at your textbook on page 238. So during the formation of the gametes, any allele for the seed shape can pair with any allele for the seed color. Okay, I repeat. During formation of the gametes, any allele for the seed shape can pair with any allele for the seed color. Alright, so that show the Mendel's second law, or law of independent assortment. Yeah, during the gamete formation, each member of a pair of allele can combine with randomly with any allele from another pair of allele. Okay, so what is the summary of the Mendel dihybrid experiment? Okay, the summary first. New combination of the characteristics are produced in F2 generation. In F2 generation, namely constricted yellow seed and round green seed. Yeah, okay, so in the F2 generation, we can see the new combination of the characteristics. Okay, what are the new characteristics? They are constricted yellow seed and round green seed. Okay, second one, the two characteristics that refer to the seed shape and seed color are combined in F1 generation, but later they separate and react freely in F2 generation. Okay, so why that is happen? Because in the gamete formation, any allele for the seed shape can pair with any allele of the seed for the seed color. Right? Okay, so that is shown by the Mendel dihybrid experiment. So this diagram show the formation of the gamete in the meiosis. So this is referred to the metaphase 1. So what can we see from the diagram here? What can we see? The each allele from a pair of alleles can combine randomly with any allele from any pair from another pair of allele. Alright. So the diagram show the law of independent assortment. Okay, I repeat, the law of independent assortment stated that during the gamete formation, each allele from a pair of alleles can combine randomly with any allele from another pair of alleles. So it will produce the different combination of alleles in the gamete. Okay, so that's all for now. For the 11.2 dihybrid inheritance. Okay, so we will do the exercise more in this topic. Alright, because we have to know how to construct a dihybrid cross. Okay, so that's all for now. Take care, everyone.